So, just to introduce this, the conundrum I'm talking about won't be the conundrum of me losing my speech in a speech workshop, but hopefully the conundrum has more to do with different synchronizing and importantly desynchronizing behaviors in the service of memory. All right, is this working now? Good. Okay, good. Okay, so I'm interested in episodic memory. That's the poison of choice, I suppose, for me. And we need to take a few moments and think about what actually is episodic memory, because it is a very specific type of memory that can be distinguished from other forms of memory, like implicit memory, like working memory, and so on. So I'd like to introduce this with a bit of an autobiographical example. So when Lars asked me to join the band, I dusted off my bass, right, which I haven't played in many, many years. And I was time warped back to like 17 years ago, where I did play lots of music. And lots of memories were triggered by me picking up the bass again. And these were episodic memories because I could pinpoint them to a particular location at a particular time. And these were highly multisensory memories, so involving visual reinstatements of locations, of gigs we played, but also obviously reinstatements of sounds and songs and all of that. And I could vividly replay these in front of my mental eye. And the question, the very non-trivial question, is how does the brain afford us to get these memory in and retrieve them and replay them? And it's obviously, it's got to be a very complex process that allows the brain to do this, but I start with a simple assumption where I assume that there are at least two processes going on. And one is the brain needs to have a means of representing this information in all the different cortical areas that break down all, a complex stimulus into the different parts. And the second function is it needs to have some form of glue, some form of binding that holds all these different informations together. And how can we square this with brain oscillations? How could brain oscillations afford the brain the necessary computations to do this? Or how could brain oscillations be a signature thereof, if you want to be more conservative? So we put forward a framework which ultimately has to be wrong, I guess, but it affords us, or what it provides us with, is some very specific predictions, and we can go about and test these predictions. So here's the framework. We basically take complementary learning systems that assumes that there is a division of labor between the hippocampus and the neocortex. And this division of labor is such that the neocortex mainly represents this information, and the hippocampus glues information together because it has a very sparse sort of representation. And what I think is going on is this division of labor is visible in opposing desynchronizing and synchronizing behaviors. So how does this work? So say you meet a particular person at a particular place, and you happen to go for dinner with Jennifer Aniston here in your Italian restaurant, as it so often happens, then surely your cortical neurons get very excited, and a whole lot of them. What we will see if we record the EEG at that point is a huge alpha-beta desynchronization. We've seen many examples in Ole's talk, which I didn't see, but he assured me he showed some desynchronization. Vittoria showed very nice examples. So it's a huge signal, right? You can't possibly miss it. And I guess what we know is that this power decrease increases firing rate. So I don't think that's very controversial. But what I think what this power decrease is also doing is it affords the system some sort of flexibility to code information. So I think this power decrease is a signature, or even the computation of representing information. Now, eventually, this information will trickle down to a handful of neurons in the hippocampus, which have sparse representations. That's something we learned from Rodrigo Quiroga's very impressive work showing Jennifer Aniston neurons and also other neurons. So these neurons, because they're only a few of them, and they are very abstract abstractly hold the representations. And because they're in the hippocampus, which is highly interconnected, of course, they can do the gluing job. And my assumption is that this gluing function is seen in synchronized theta and gamma oscillations. So this framework is not a framework where I say, you know, is any particular oscillation related to memory? Is theta doing memory? Is alpha doing memory? Is beta doing memory? All of these oscillations are doing memory, right? What I'm assuming is that particular oscillations are a signature or even the computation implementing a particular process. Okay, so now this framework makes some specific predictions, and I first focus on the desynchronization and information part prediction that we tested in a recent paper by Sebastian here. So what we did is we let subjects encode dynamic stimuli, a movie, and then later on we showed them a word, right? So this is not still images, it's an actual movie. And later on, we gave the subjects the word, and they had to mentally replay the scene. And we not only wanted to do that in the visual domain, because I think the memory field is very much vision-centric, and I think that's wrong. We should be doing all sensory modalities, because we have episodic memory for all of these. So we also included a condition where we have subjects link a sound with a particular word and then reinstate the sound. So the question that we're asking here is basically, can we get some form of decoding at the encoding part in order to tell which out of 4 particular movies or 4 particular melodies a subject is reinstating? So what we hunt here for is a signature during encoding that reappears here during retrieval. And this signature is not, as an fMRI person would go about, a spatial signature. It is a temporal signature. And there's obviously lots of frequency bands and locations. The computational space is just enormous. So we concentrated on the frequency band that shows the highest desynchronization. That is the strongest power decrease during the successful retrieval. And that happened to be 8 Hertz for both modalities. You can see here these power decreases, they're huge. You just can't possibly miss it. Any cluster correction will pull this out. And so why do we go for the power decrease? And the reason why we go for the power decrease is because what I think is going on, a power decrease renders a signal less stationary, so it's more deviating from a sinusoid, and that's trivial to show, and we did that. But this non-stationarity renders the signal flexible. It gives a coding space for a particular stimulus. And if indeed these power decreases code information, we should be able to utilize this, these squiggly lines, if you will, to tell which stimulus a subject is reinstating. So that's the more or less complicated computation we did. We take a squiggly line from the encoding phase, a temporal trajectory of 1 second, fitting 8 cycles in 8 Hertz, and then we slide this over the retrieval period because we don't know exactly when this pattern reoccurs. Right here in encoding, this pattern occurs right after stimulus onset, and then here we have a similar pattern, a similar sort of phase reset. So at this time point, this sliding window should match this, and we have this pattern occurring here. So we can calculate, straightforward phase synchronicity measures, similar as you would do to calculate connectivity between regions, but here we're calculating connectivity between squiggly lines measured at different time points. And we can contrast that between the retrieval of the same stimulus and a shuffled version thereof, get the difference and get a content specificity index, if you will. And this works like a charm for encoding. I mean, Christoph Kaiser has shown quite some data, and we see the same. We did this in EEG, source localization, and works beautifully. Visual cortex, auditory cortex, perfectly pulls it out, so I don't gonna show this data. What I show is the reinstatement of these patterns. And so the short version of this is it works. We can decode the visual memories, we can decode the auditory memories, and we source localize these to higher, order visual areas and maybe higher order auditory areas. We can also look at the time course. Visual information is reinstated rather quickly, whereas auditory information takes a bit more time, which, we didn't have a specific story about that. Maybe you guys can help us figure out why that's the case. And to then finally link it back to the power decreases, what we did is we took these ROIs and then looked at the power in those regions of interest. And if it is the power that affords this reinstatement of the memory, then we should see an interaction such that power is lowest in the visual ROI when the visual information is reinstatement compared to when an auditory information is reinstated, and vice versa, and that works. So there was a great poster yesterday by Johannes. I hope you checked it out. He applied a similar algorithm, actually the same algorithm in speech and working memory. Okay, so that was the desynchronization part. Now I will focus a bit more on the synchronization part and how we can test this. So there is obviously a hell lot of literature linking theta to LTP, LTD. I guess it was triggered by an initial study, an in vivo study by Pavlidis, 1988, that was replicated and sliced by Huerta and Lisman, showing that particular theta phases might represent time windows for LTP, LTD. So the long-term potentiation and depression, of course. So the idea here is that if you want two regions that represent maybe two features of a memory to form an association, they need to be synchronized to the LTP phases in theta. And we have some correlative evidence for that using MEG and localizing hippocampal activity. And so in one study we found that indeed there is a 4 Hz specific signature of theta phase where gamma sits on particular theta phases when an association between a word and a movie is happening. We replicated this 4 Hz specific effect in a recent study together with Christian Döller in this current biology paper by Alex Bacchus. And it always was 4 Hz, which I found interesting because that gives us something to play with in a more causal manner. So the thing that we wanted to do here is reverse the axis, so mess with the synchronization and see what it does to memory. And see whether that is frequency specific. So how did we do that? The experiment that was run by Andrew Clouter together with Kim Shapiro relied on the following assumptions. So say, for instance, we have a multisensory memory. You see someone talk or you watch a movie and it has sounds. And so we have an auditory in the visual region. If an association between the two has to be formed, then presumably the hippocampus needs to synchronize these two guys. So when we then derive these two regions in an asynchronous manner, then that should be bad for memory because they never arrive at the same theta phase, right? If we drive the two regions in a synchronous manner, it should be good for memory. So that would be a straightforward prediction. So how did we do that? Obviously, we can't use optogenetics in humans, but we can show funny movies like this. Let's see whether that works. Okay, so that is a short example of few multisensory stimuli we used. That looks a bit like an arts project going wrong, but actually what we do here is we drive the visual and the auditory cortex in different synchronizing and desynchronizing ways. Actually, you've seen here examples of synchronized and desynchronized auditory visual stimuli. You probably couldn't tell it, which is perfectly fine because the subjects can't. To stop this here. Okay, and later on we test the memory. We just played a sound and we asked the subject to tell us which out of a couple movies was related to that. So first of all, we need to do some sanity check. We obviously need to control for the latencies. Christoph has mentioned that very nicely in his talk. We need to adjust for the latencies between the visual auditory systems, so we did that. And we measured the EEG to see whether we got it right, and the short answer is yes, we got it right. So when we present the two stimuli in sync, we get a nice synchronization between an auditory and the visual source. And in 90 degrees, it's at 90 degrees, and then it goes around the unit circle. So we have these 4 different phase conditions. And then the more interesting question is, does memory vary as a function of this phase difference? And it does. It's one of these things where your PhD student goes to your office and says, you know, Simon, the crazy ideas of yours, it actually worked. Doesn't happen too often, I don't know, at least to me. So the memory performance is much better in the 0 than in any of the asynchronous conditions. So we lump these together, call them asynchronous and call this synchronous. Then we can plot it like this. And then obviously, if this is a mechanism specific to theta, it should only occur at theta, right? So when we do the same experiment in this lower frequency and in a higher frequency, we should not find this effect, because that would link it really to theta and possibly LTP, and would not just be about audiovisual synchrony. And so we ran a second experiment, different set of subjects. We again found this 4 Hertz difference between synchronous and asynchronous. However, there was no difference whatsoever in a lower frequency, 1.7 Hertz, and at 9.8 Hertz. We chose these funny frequencies because of avoiding harmonics and subharmonics and stuff. So that is a frequency-specific effect, which is why we called it the theta-induced memory effect. Okay, now then in another set of EEG studies, we wanted to relate the variation on a single trial level in the phase difference between auditory and visual cortex to memory. Because obviously when you average across your trials, you will see, you will pull out the phase difference that you put in essentially, but the brain doesn't stupidly follow that. On the single trial level, there's a hell lot of variance. And the question is, can we relate this variance to later memory performance? So we did that in this EEG study, and we again replicated the behavioral effect. It like always checks out. We have run 5 replications so far. It's pretty robust, which is good, I think. And then we also replicated the phase difference. The auditory source and visual source are phase locked in the 0 degree condition and the 180. So we only ran these 2 conditions to maximize our power here. Now the more interesting question here is, when we split our single trials based on later memory, what happens to the phase synchronicity in those 2 regions? So in the 0 phase degree condition, I guess not so surprisingly, we find that later remembered items synchronize more to this optimal phase, and later forgotten items show more variation. You see here a slight difference in the mean resultant length of the vector. Now the more interesting prediction is what happens for the asynchronous. In other words, do we find some, say, rebellious trials in the asynchronous condition that withstand the non-optimal entrainment and have some clustering at around 0? And the answer is yes, we do. So for later remembered items, we here find actually less phase locking to the entrainment, but and a bit of a clustering here at around 0, which presumably could be here responsible for these items being later remembered. And we find a stronger phase locking to the 180 degree when the items are missed, and we can test this more formally by using pairwise phase consistency, and we see this full pattern of interaction. Okay. So these line of studies, we cannot sort of study the desynchronization part and the synchronization part in isolation, but obviously it somehow has to come together. And the question is, how does it come together? It can't be, a very simple scenario here. It has to probably be a bit more complex. And in order to understand what these mechanisms are, I turn to computational modeling. with the help of Howard and George here to get some very clear-cut predictions from the model and get some idea how what is going on. So in this model, which is very, very simple, and we're ignoring, all the different subregions in the hippocampus here, which because we just wanted to make the model as simple as possible in order and account for as much as possible data. Okay, so what do we assume here? We have a neocortex that is driven by alpha, okay, in the resting state or when nothing's going on, and we have a hippocampus that we drive with theta, and we assume that learning occurs in the hippocampus, not in the neocortex, and this learning occurs via spike timing dependent plasticity that is modulated by theta phase, such that we have an increase in synaptic connections in this trough here, and a decrease of synaptic connections in this peak here. So in the resting state, alpha drives the neurons in the cortex, theta drives the neurons in the hippocampus, and when there is an input, these guys get excited, alpha decreases, and these guys move forward in phase. And they move forward in phase because their threshold is lowered, right? And this allows the hippocampus to separate units that are non-relevant from the units that are relevant and selectively strengthen these. I'm good. And we tweak the model such that it would fit some single unit data, because we have single units obviously in the model, using the study by Eisen, where they showed that a particular neuron that would prefer to fire for Jennifer Aniston here, but not for the Eiffel Tower, can change its tuning curve after the subject has learned an association between the two, which is the green line here. So our model can model this. So we have two sets of populations in the neocortex, the hippocampus, and before the learning, the set of these neurons won't fire to the Eiffel Tower, but after learning, they do. So that is the thing that I want you to focus here. During encoding, obviously, both fire like crazy because we show both stimuli. Now, what happens with regards to this theta phase is that this neuron that would not be interested in the Eiffel Tower before fires at the theta peak, the most excitable phase, which is related to long-term depression. And then during encoding, it moves its firing phase. And here we only count the first spike, okay? Because actually the neuron will fire like a hell lot throughout the cycle, but we only count the first spike here. And that first spike occurs in this theta trough where LTP occurs, which is how the model learns. And then we can see when we later present the non-preferring stimulus and a sort of effect of that learning occurring by the theta phase shift. Now, another thing that the model does is it shows this flip-flap pattern of 5 minutes. All right, yeah, I can deal with that. Of power decreases in alpha neocortex and power increases in theta. And so we can test this, and there was a beautiful poster by Ben Griffiths, which you may or may not have seen, where we correlate the power in the neocortex with the power in the hippocampus, and we can see a negative correlation, a relative negative correlation when a memory is being formed, such that we have a decrease of power in the neocortex occurring with a theta power increase and also a gamma power increase. So that's proof for this desynchronization, synchronization behavior. And another very exciting thing we can do is we can record single units. We have recorded 8 patients so far, and we find single units, and some of them even are phase locked to theta in the hippocampus. This is one example. And we can find these concept neurons. So this is just an example for a neuron that fired to Theresa May. We found this neuron on Sunday. On Monday, it was no longer there. Strong and stable leadership. I don't know. We found a neuron firing to the Sydney Opera House. So we can then utilize this, ask the patient to make an association between these two stimuli, and then specifically test this prediction of theta phase precession. Hopefully for the next meeting I will have some data to show that. So to conclude here, what I tried to convince you of is that we have different sort of neural computations going on with a desynchronized alpha beta that might allow the system to flexibly code information, and the synchronized theta gamma in the hippocampus that is the glue. So the last 2 minutes, I just want to show you some data that does not fit with the model, because, you know, we always try to make everything coherent and stuff, but, you know, data is not always coherent, so, and I want to share this frustration with you. So this is an example where we did spatial memory encoding using the method of loci, which probably ticks every box that gets the hippocampus and should get the theta going, right? It's spatial cognition, it's associative memory, and all of that. And in the fMRI, we find a huge bold signal increase in the medial temporal lobe using this technique, and also contrasting later remembered and not remembered items. However, in the EEG, we see a huge theta power decrease. So theta power desynchronization in MTL during a spatial episodic memory task. So that doesn't obviously square perfectly yet with the model. Another thing that I wanted to show, especially since Nancy is here, is we found this neuron that is like a beta clock. So the autocorrelation shows this nice peaks at 67 milliseconds, and obviously when we do an FFT here, we get a huge peak at 16 hertz. When we show a stimulus and let the patient do some memory, it sort of breaks from this synchronization. So that's perfectly in line with the desynchronization idea. However, this neuron is in the hippocampus. So I don't know what's going on there. Okay, so that keeps us busy, I guess, and try to figure out what's going on there. So thank you for your attention. Thanks to these great people. I'm really fortunate to be able to work with, the funding, and most important, all the patients for their time. Thanks.
Usual suspects, yes. I just wanted to make a critical remark regarding your first experiment. I don't think that because you have a pattern during the retrieval that can be decoded, it tells you anything about that this is causal for retrieval. And we actually have data showing that you can have beautiful decoding from regions that have absolutely nothing to do with the process you're interested in, and it can just be, you know, byproducts, correlations of, yeah. So we have, I think it joins a point that Jonas raised yesterday. Hello, Jonas. So it's important, I think.
I mean, yeah, it's correlation. It's not causation, but, you know, correlation is not nothing. I think we can learn something from there. Obviously, what we need to figure out is what exactly is driving the reinstatement of those patterns. What are the mechanisms that trigger internally, purely memory-driven, these phase resets? So yeah, there's some work for us to be done.
I really like your temporal pattern analysis, the first experiment also, and but what I'm concerned about is during the encoding and during the retrieval, you don't expect you expect to have a compression of the information in between the two.
Yeah.
So what's your logic behind using this temporal pattern analysis?
So this is exactly the question that we address in a follow-up experiment. Let me. Oh, where's this? Yeah. Okay. So yeah. So that was actually the initial question we tried to address. What is the temporal compression? Because obviously there has to be temporal compression in memory. And what I wanted to see is sort of a trajectory, you know, being above the diagonal if we plot the encoding time against the retrieval time. The data did not play ball in this paradigm, because we found during encoding that the most informative patterns happen right at the onset of the movie. They were a bit more sustained for the auditory, but there overall the effect was weaker. So what we're doing now is we're showing a complex movie that consists of 3 scenes. For each onset, we get a temporal signature, and then later we ask the subject to retrieve these and then we can get the exact temporal compression. Yeah, and I mean, so far that is working, and we'll probably have some answers to that next time.
I have one question regarding your, because you're presenting a movie and then the word, but then when you are decoding or when you're trying to reinstantiate the predictions or the memory, you're doing it the other way around, right? And one of the things that we know from theta phase precession is that if the rat runs in one direction versus in the other direction, it's not the same memory, right? So I'm trying to understand, like why your memories work forward and backwards. When they should go forward.
The reason for this was purely methodological. We wanted to make our lives as miserable as possible, finding temporal patterns. Because if you would show the word before the movie at encoding, right, then you can think about some temporal autocorrelation that lingers on, that represents some features of that word, or you have the word in your working memory, and then your temporal pattern similarity will pull that out. And that is not directly due to a memory retrieval, but it's more, a signature of working memory maintenance. So that is why we switched it. So we show the movie, we get the temporal pattern, the footprint of the movie at the time point where the word, which we show late at retrieval, is not shown, and the subject has no idea about which word we show.
I mean, I understand your method, why you did it methodologically, but I'm puzzled about the mechanism that entails. Right, because a forward prediction, like, in this case, you would expect that A predicts B, but not that B predicts A. That's what the rat hippocampus, would tell you. Right? So that's why the memories that going backward and forward are not the same.
I don't see a problem here. You have paired associative learning. You can cue both memories in both ways. I mean, well, I can see where this is a problem when you think all that is happening is spike time independent plasticity, and if that is the mechanism, then you have asymmetry. You can cue the memory with one, but you can't cue it with another, which we are testing with this entrainment paradigm using gamma oscillations. Because there we can, if it's spike time independent plasticity, if we give one item a lead before the other, then we should get this asymmetry. But in our model, which learns by this theta phase mechanism, this is not a problem. We have, we can cue the memory each way. And each way it works.
Any more questions, complaints to the management? If not, then thank you, Zuma, for